You know that feeling, right? That sense of whiplash you get from science news? One day, a headline is telling you a certain food is a miracle cure, and the very next day, another one warns it might kill you. It is super frustrating and honestly, confusing. So in this explainer, we are going to get to the bottom of why this happens, and more importantly, what you can do about it. I mean, you've absolutely seen this exact thing before. On Monday, coffee is the key to living forever. By Friday, you're convinced every sip is a huge risk. This isn't just in your head, it's a real common experience. So what's going on here? Is the science itself constantly flip-flopping, or is there something else happening behind the scenes? And if you've ever felt that letdown, you are definitely not alone. This quote just nails that feeling, doesn't it? We get promised this world-changing discovery in a headline, but then when you dig into the actual article, the reality is, well, a lot less exciting. This is a recognized problem, and we're about to pull the curtain back on exactly why it happens so often. Okay, so here's our roadmap. We're going to start with that headline roller coaster we all ride. Then we'll trace the story backward through what's called the distortion pipeline. After that, we dive deep into a huge issue inside science itself, the replication crisis. We'll look at why findings don't stick, give you a toolkit to read news like a pro, and then wrap up with the good news, how science is fighting back.
You know, this constant back and forth does more than just confuse us. It can make us cynical. When it feels like experts are changing their minds every other week, it's really tempting to just throw your hands up and tune it all out, which means we might miss genuinely important health advice. So to understand this, we need to follow the story all the way from the lab to the headline.
The best way to think about a scientific findings journey is like a giant game of telephone. You know the one. The original message starts out complex, filled with nuance and caveats, but at every single step on its way to you, a little bit of that nuance gets shaved off, and a little bit of hype gets added on. Let's break down those steps.
And here they are, the 4 key stages where things get warped. It all starts with the study itself, right? Researchers, and especially their funders, might, consciously or not, design a trial in a way that's more likely to give them the positive result they're hoping for. Then, scientific journals, which want to publish exciting, groundbreaking work, are more likely to accept those positive studies. Next, the university or company involved blasts out a press release, often puffing up the findings and burying the limitations. And finally, a journalist, who's likely on a tight deadline and not a science expert, grabs the most dramatic part of that press release to write a headline they know you'll click on. See? Each step adds a little more distortion.
But here's the real twist. It would be easy to just blame the media, but that's not the whole story. Not even close. The problem actually goes much, much deeper, right into the heart of how science gets done. For a while now, there's been this quiet, growing issue bubbling just under the surface, and today, it has a name: the replication crisis. So, what in the world is the replication crisis? Well, at its core, it's the discovery that many scientific studies, when other independent scientists try to repeat them, just don't give the same results. And that is a huge deal, because replication is supposed to be the absolute cornerstone of science. It's how we check each other's work and verify that a finding is a real thing and not just a random fluke. Now, this chart really puts the problem in stark relief. A massive project tried to replicate 100 different studies from top psychology journals. Out of the 97 original studies that claimed to find a significant result, guess how many they could actually reproduce? Only 35. Just over a third. That is a shocking number, and it sent waves through the scientific community. And before you think, oh, that's just a soft sciences problem, it is absolutely not. This crisis hits the so-called hard sciences too. For instance, researchers at the biotech firm Amgen tried to replicate 53 landmark studies in cancer research. They could only reproduce the findings of 6 of them. That's a replication rate of 11%. We're talking about research that life or death treatments are built on.
Okay, so why? Why is this happening? Now, it's rarely about outright fraud or scientists with bad intentions. It's more about subtle systemic pressures and common research practices that can unfortunately lead to a lot of shaky findings being published. Let's break down some of the main culprits.
First up is something called publication bias, which is also known as the file drawer effect. Just imagine 10 research teams all test the same new drug. 9 of them find it does absolutely nothing. One team finds that it works. So which one of those studies do you think gets published in a flashy journal? The exciting positive one, of course. The other 9 studies get quietly tucked away in a file drawer, never to be seen. So anyone reading the literature gets a completely warped view of the evidence. Next, we have a practice with a kind of silly name but very serious consequences: p-hacking. There's a famous saying in science: if you torture the data long enough, it will confess to anything. That's p-hacking in a nutshell. It means slicing and dicing your data, trying different analyses, or maybe adding a few more subjects until you can get a result that looks statistically significant, even if that pattern is just meaningless, random noise. And this one is a huge contributor: low statistical power. It's a fancy term for a simple problem. A lot of studies are just too small. Running a small study is like trying to figure out if a coin is rigged by only flipping it 5 times. If you get 4 heads, you might think it's biased, but it could easily be just random chance. Small studies are way more likely to produce dramatic-looking results that are really just flukes.
And to put a number on that, the accepted standard for a study to have adequate power is 80%. That means it has an 80% chance of detecting a real effect if one actually exists. But when scientists went back and analyzed the published research, they found that the median power in many psychology studies was under 40%. That's worse than a coin flip's chance of finding a real effect, which goes a long way to explaining why so many findings just vanish when someone tries to replicate them.
So, I know this all sounds pretty discouraging, right? But it's not hopeless. In fact, just by understanding these problems, you can arm yourself with the right questions to ask. Think of this next part as your own personal, practical toolkit for becoming a much, much smarter consumer of science news. All right, so how do you spot a flimsy story? First, be extremely skeptical anytime you see the word breakthrough. Real ones are incredibly rare. Second, see if the article quotes independent experts, people who weren't involved in the research. A good story will have them. Also, ask yourself, was this a study on actual humans, or was it just in mice or cells in a petri dish? And finally, did they measure something that really matters to your health, like preventing heart attacks, or just a proxy measurement, like lower cholesterol? Those are very different things.
And you have to keep an eye out for these red flag phrases. Words like linked to, could prevent, may help. These are often clues that a finding is super preliminary and very uncertain. When you see these words, your internal alarm bells should start ringing. They're a signal to take the claim with a giant grain of salt.
And this timeline just puts everything into perspective beautifully. Real science isn't a single eureka moment. It is a long, slow, grinding process. A true medical advance can easily take 15 years or more to get from a discovery in a lab to something your doctor actually recommends. Any single study you read about is just one tiny, tiny step on a very, very long road. And now for the good news, because there is good news. The scientific community is not taking this lying down. In fact, this whole crisis has sparked a huge internal reform movement to make research more transparent, more rigorous, and more reliable. So let's take a look at how science is fighting back. Some really big, important reforms are starting to take hold. One is called pre-registration. That's where scientists have to post their entire research plan online before they even collect their data. It makes p-hacking pretty much impossible. Some journals are also moving to a result-blind review process, where they decide to publish a study based on the quality of its methods alone, not on whether the results were exciting. The open science movement is pushing everyone to share their data and methods so anyone can check their work. There's even a whole new field called metascience, which is literally the science of making science better. So when you see all these debates and corrections, it's not a sign that science is broken. It's actually the opposite. It is the sign of a healthy system working as intended. This is what science correcting itself looks like. It's just that now it's happening out in the open for all of us to see, and it can be messy. So that really leaves us with a final question. This process of self-correction is absolutely essential for progress, but it isn't fast. And in our world that wants instant answers, the real question is, are we patient enough to watch it work?